you know, out of the body. And so your colon just gets huge, megacolon just chock full of feces. And the same thing can happen in the esophagus, where, you know, the smooth muscles can no longer contract. So the esophagus can't help propel the swallowed food down to the stomach. And can you imagine a horrible way to die? Yes, Grace. One of the cadavers here has a megacolon? I didn't know that. Do they think it was from Chagas disease, or is it another?
It's really old one.
Okay.
But yeah.
Okay. Wow. Oh, that'd be so cool. Maybe the A&P folks will let us come down and look at the cadaver that has megacolon.
Oh no.
Oh no! Oh yeah, okay, or not. We can make it optional. We can make it optional. Okay, but certainly, you know, horrible way to die, right? So that's called mega.
It's like, oh no.
And mega esophagus. Mega esophagus. So we can see the parasites that destroy cardiac muscle and the smooth muscle of the gastrointestinal tract. Yeah, Bianca.
Why do you call it peripheral blood sample?
Oh, peripheral blood sample. The peripheral veins and arteries versus, help me out here, Alicia. So central, like aorta, vena cava would be considered like not peripheral, central. Is that right? You guys, MP, help me out, you guys. Okay, good. Peripheral. So when we go for a blood sample, you know, taking it from a vein, usually a vein, right? Is this right? You guys help me?
Yeah.
Okay. Considered peripheral, right? And the reason that's important is that some of the parasites, when we start getting into the lymphatic filarial parasites, they only circulate in the periphery at night when mosquitoes are biting. And during the daytime, they stay more in the core portion, the blood vessels in the core portion of our body. So it becomes important. You're going to take a peripheral blood sample of patients at night to detect the little filarial worms. And this is on a whole other topic we're going to hit next week. But just showing that some of these parasites, they actually have variations in if they're circulating in the periphery or if they're hanging out in the core, the blood vessels in the core portion of our body. Okay, yeah, that was a great question. Every semester I learn from you guys. Every semester I'm like, wow, I never thought about that. So, okay, all right. Okay, so those are our flagellates. Okay, the flagellates. So remember, you guys, the Giardia is in a fecal sample demoscope. The Trypanosoma brucei is in a peripheral blood sample. There's a demoscope. But again, I don't mean to discourage you from looking at your own slides. But sometimes, like the fecal ones, they're hard to look at. Okay. The next one we're going to do are the ciliates. And we don't have as many examples on these other ones, so it'll go faster, you guys. So the next group are the ciliates. And we just have two examples of ciliates. Okay, and maybe I can just get them right here. Yes, I can. Okay. And what I will try to do, you guys, this weekend is I'll try to put this little table on D2L. And one of your colleagues, Shireen, yesterday mentioned that Dr. Holland, I think she wouldn't mind if we did this. And Dr. Holland's lab module on D2L, Dr. Holland has some absolutely gorgeous handouts for her lab. And one of your classmates said one of her handouts, it was this beautiful table kind of breaking down the protists. And what was so lovely about it was that she had included little photomicrographs. And your colleague held it up, and it looked absolutely gorgeous. So I'll try to make one for us, but in the meantime, you might want to download and print the one from Professor Holland. She's just got some beautiful, beautiful handouts. And I don't think she'd mind if you downloaded them and printed them. Okay. All right. So we did the flagellates. Now we're going to do the ciliates. And ciliates are organisms that have, as you might guess, eukaryotic cilia. And the eukaryotic cilia, the structure is identical to the flagella. They are covered with cytoplasmic membrane. They have a 9+2 arrangement of microtubules. The difference is usually the cilia are shorter than flagella, and usually the organism or the cell has lots and lots of cilia. whereas with flagellated organisms, usually they just have a few flagella. Okay. So our example of free living is another really neat organism called Paramecium. And this might be one of the protists that you guys have seen already, maybe in previous biology classes. So we've got a model of Paramecium, and it's this little guy right here. Right? Now, if you take a look at the models here, you might ask, why did we put two different keys to the Paramecium model? The top one matches our model here, but you'll see there's a second Paramecium model below. And the reason I put the second one there is the artist on this model left out something really important. What's missing? The cilia, right. So I just wanted to put that second key there to remind you that we have a naked Paramecium here. It looks like it got sheared. Probably just the cilia broke off all the time, so they decided, oh, we're not even going to put the cilia on this guy. Okay, but do remember that in reality, the Paramecium would be totally covered with thousands and thousands of these short little cilia that likewise are beating back and forth. Okay, so let's take a look at the list on our little Paramecium and make sure we get everything. Okay, now, we've already talked about how the cilia would be located on the outside of the Paramecium. So the function is what? Motility, right? But there's a second function for cilia and ciliates. The cilia line this invagination called the oral groove in the gullet. The cilia are going to be to create water currents that will draw food particles down the oral groove in the gullet. And here, right at the very end, we're going to have a process called endocytosis occurring, or phagocytosis. Okay? This is when the food molecule is enclosed in a membrane-bound vacuole. Okay? So these little light guys right here, we're going to call these food vacuoles. Food vacuoles. Now, the way the nutrient would be digested is when the food vacuole is formed, another membrane-bound organelle called a lysosome is going to migrate over, fuse with the food vacuole, and dump hydrolytic enzymes inside the phagolysosome. And those hydrolytic enzymes will break down the proteins, nucleic acids, the sugars, the lipids. The smaller nutrient molecules will be absorbed into the cytoplasm of the organism. And what will remain will be waste. And to me, you guys, these look like little fecal pellets, right? Okay, so these are the waste vacuoles, And just like us, these organisms have to rid themselves of this waste. So the waste vacuoles will migrate to a dedicated portion of the cytoplasmic membrane, right up here. The membrane of the waste vacuole fuses with the cytoplasmic membrane, and the waste is expelled in a process called exocytosis. Okay? So let's just go through it really quick, you guys. What is the process occurring at the tip of the pointer?
Endocytosis.
You could say phagocytosis. If you said endocytosis, that would be fine too. Identify the structure at the tip of the pointer. Food vacuole. Identify the structure at the tip of the pointer. Waste vacuole, just there's a number of them here. Okay. Identify the process at the tip of the pointer. Exocytosis. Dynamite, you guys. Excellent. Okay. Okie dokie. Now, I'm sorry. Okay.
So there's only one of those things for exocytosis?
Okay, that's a good point. On your models, I think it's called the anal pore. And the way I've read it described, it's a dedicated portion of the cytoplasmic membrane. So I'm presuming that in this general location, that's where the waste vacuoles fuse with the cytoplasmic membrane. Now, whether it's the identical place over and over again, I don't know. But it's this general region, I believe, is where most of the waste vacuoles fuse. Yeah, it's kind of cool. In a unicellular organism, we have dedicated cytoplasmic membrane for waste expulsion. That's kind of cool. Yeah, yeah.